Good morning to all. Welcome to the Diamond Group of Institutions NEET and K-CET online classes. Today's class is mineral nutrition. In that some portions are select the incorrect match of essential elements. So you know that essential elements, which are the elements are required for the plant growth and development. So based upon that requirement only, essential elements are identified as a 17 essential elements. Out of the 17 essential elements, based upon its functions, we have to identify the incorrect match. See, they're given as a manganese. A. Option A, manganese. Here, the match it is structural component of chlorophyll. B, calcium, component of middle lamellae. C, zinc enzyme activator. And D, component of iron, component of ferridoxin. So here, option A, manganese they given. Manganese is a major component into the photolysis of water. It's involved into the photosynthesis, but it's not involved into the structural component of chlorophyll, because when we see the structure of chlorophyll A and chlorophyll B, center magnesium, sorry, central magnesium elements will be going to present. C55, 72, 5 and 4 mg. And it will going to connected by the 4 nitrogen atoms. 72, 5 and 4 mg. So when we see as here, center single magnesium element will be going to present. Magnesium is the component of the chlorophyll pigments. Here, instead of the magnesium, they are given as a manganese. Manganese is involved in a photolysis of water according to the Hill's reaction. In the presence of light, this water will going to split into 2 protons, 2 electrons and half oxygen molecules. This is called as a photolysis of water. In this reaction, manganese element will be going to involve. So for this match, they given as a structural component of chlorophyll. That structural component of the chlorophyll is not a manganese, it's a magnesium. And see now, calcium, component of middle lamella. So when we see the cell wall of the plants, plant cell wall, it's contained as a middle lamella, and firstly formed cell wall is a middle lamellum only. That middle lamellum is contained as a calcium peptide and magnesium peptide. So function of this middle lamellum in a cell wall. So these are the middle lamella can act as a cement like structure in between the two cells. And option C, zinc one of the major microelement. It is used into the activation of different types of the enzyme. The different types of the enzymes are alcohol dehydrogenase enzyme. And option D, they are given as iron, component of ferridoxin. So iron also involved into the photosynthesis. Iron also involved into the majorly ferridoxin. In the photosynthesis, when we see into the cyclic and non-cyclic electron transport system, is here cyclic non-cyclic electron transport system. Finally, the 2 electrons will be not received by the ferridoxin. The ferridoxin is made up of by the iron molecules. So out of the given options, incorrect combination is manganese. They given as a structural component of chlorophyll, but it's not a structural component of chlorophyll. It is involved into the photolysis of water. And next question, plants require which one of the following for the formation of ATP? You know that ATP represent as adenosine triphosphate. So out of the given molecules or out of given elements, which of the elements are majorly involved into the formation of ATP? See, they are given as nitrogen and calcium. Calcium it is used into the formation of cell wall and also the formation of spindle fibers at cell division. And option B, they are given as nitrogen and phosphorus, nitrogen and copper, and potassium. Potassium is involved into the that is opening and closing of the stomata that can maintain the turgidity of cell. And coming to the copper, it is involved into the different types of the redox reactions and also involved into the activation of the enzyme. See now, here out of the given 3 options A, B, C. Nitrogen they are given. Nitrogen one of the macromolecules of the all living organisms. Here nitrogen will be going to presence into the all living organisms in the form of nitrogen bases. So you know that DNA and RNA, that is genetic material can made up of by the different types of the nucleosides and nucleotides. The nucleosides and nucleotides are they having nitrogen bases as purines and pyrimidines. So those are the purines and pyrimidines are adenine and guanine with all as a purines, cytosine, thymine and uracil with all as a pyrimidines. So now these are the adenine and guanine and cytosine, thymine, uracil. These all are with all as a nitrogen bases only. So when we seen as a ATP, ATP abbreviated as a adenosine triphosphate, it's the energy currency of cell. Adenosine triphosphate. So when we seen as a adenosine, adenosine means adenosine is a nucleoside. So these are the nucleoside. It is made up of by sugar molecules. The sugar molecule connected into the nitrogen base of adenine. So sugar plus nitrogen base of adenine we call as a adenosine, and they given as a TP represent as a triphosphate, means it's contained as a fifth carbon atom, 3 phosphate molecules. So adenine and phosphate molecules and sugar, these are the nitrogen base and sugar molecules we call as a nucleoside, and by the presence of phosphate it's called as a nucleotide. So ATP is a nucleotide. It's contained as a 3 phosphate molecules. So that ATP, adenosine triphosphate, it's made up by the presence of nitrogen and phosphorus molecule. So why we are not choosing to the other options here? Nitrogen nucleoside is connected into the 3 phosphate molecules. Phosphate is one of the macromolecules. So that's why ATP contained as a nitrogen and phosphate molecules only. Answer is D. And next, third one, match the following. They given as a chlorosis, necrosis, white birds, and a soil-less culture. Option A, maize, leaves, hydroponics, and a death of cells or tissues. So you know that chlorosis is a disease. The characters of the chlorosis, it is yellowing of leaves, yellowing of leaves. So by the that is lack of chlorophyll pigments in that area, like leaves will be going to becoming as a yellowing color. That's called as a chlorosis disease. It is occurred by the lack of different types of the micro and macro elements, by the deficiency of this. And necrosis. Necrosis also is a one of the disease. It's called as death of tissue. Death of tissue at net region. So that is called as a necrosis disease. It is also caused by the defect of, by the deficiency of micro and macro elements. And now you see white birds and soilless culture. First see the soilless culture. Soilless culture means Julius Van Sachs, he was proposed cultivation of the plants even though the absence of soil in the presence of liquid nutrient media, that is called as a soilless culture, that is proposed by Julius Van Sachs. It's called as hydroponics. And white birds. So out of the given options, white birds are. So here leaves to hydroponics and death of the tissues. And white birds are we are seeing in the maize plants only. So chlorosis we are seen in leaves, necrosis it's seen, or it is also called as death of tissues, white birds as a maize, soilless culture we call as a hydroponics. So 1-4 means 1D necrosis, sorry 1B, 2D, 3A and 4C. 3A and 4C, 1B, 2D, answer is a C. And next question, N2 plus 8 electrons, 8 protons plus 16 ATP molecules gives rise to ammonia, 2 ammonia molecules and 2 protons and 16 ADP plus 16 inorganic phosphate. The above equation says as. So here this equation will be seen at the time of nitrogen fixation, means nitrogen cycle. So in of the nitrogen cycle, different steps are we are discussed. Those are different steps of nitrification. Nitrification is a mechanism fixation of atmospheric nitrogen into a soil nitrogen as nitrates, nitrites. It's called as or conversion of here the nitrification says as sorry ammonia is converted into nitrites and nitrates. So this is the nitrate and this is the nitrate. Conversion of ammonia into nitrites and nitrates it's called as a nitrification, and reductive aminization it's the last step of the nitrogen fixation means nitrogen cycle. So those are the reductive aminization means fate of ammonia we can see it is. So after the formation of ammonia, ammonia is converted into alpha ketoglutarate acid, and now this of the alpha ketoglutarate acid is converted into glutamic acid. Conversion of alpha ketoglutarate acid into the glutamic acid it's called as reductive aminization mechanism. And denitrification after the formation of these after the formation of the ammonia, from the ammonia it can converted into from the ammonia or from the soil nitrogen it can enter into from the soil into nitrogen will be enter into the atmosphere, that is called as a denitrification. And option D they given as biological nitrogen fixation. Atmospheric nitrogen is converted into the soil nitrogen in the presence of living organisms, that is called as a biological nitrogen fixation. Here the equation 10 represented as two nitrogen molecules. These two nitrogen molecules are they connected together by three covalent bonds. These are three covalent bonds are they are very strongly connected together. For the breakdown of these each covalent bond, two protons are required. So that's why we are going to supply into the 8 protons, and also they will be going to utilize into the 8 ATP molecules for the formation of one ammonia. So now here at the time these two nitrogen molecules, atmospheric nitrogen molecules are getting converted into the two soil nitrogen molecules as ammonia. At the time 16 ATP, 8 electrons and 8 protons are they required. Out of the 8 protons, 6 protons only utilized, remaining 2 protons will be not releasing into the normally. Those are used for the biological processes. So the given equation is called as a biological nitrogen fixation. Answer D. Next question. Farmers in a particular region were concerned that from premature yellowing of leaves of pulse crop might cause decrease the yield. Which treatment would be most beneficial to obtain maximum yield? Here the question it is, a farmer he was got the problem, that is the problem it is plants in your crops they got getting as premature yellowing of leaf. The premature yellowing of the leaf is called as a chlorosis disease. So chlorosis disease means leaves are they becoming as a yellow before the dating of maturity. Whenever the leaves are they becoming as a yellow, their leaves will not prepare much food materials, automatically yield will be going to decrease. Two, for the increasing of the yield of crops, which is the best useful method in a given option we have to identify. Option A, application of iron and magnesium to promote the synthesis of chlorophyll. Here chlorosis disease is occurred by the deficiency of several different types of elements. Out of the several different types of the elements, so one of the element here, some of the elements are there. So you know those are out of that 8, 4 are macro and 4 are micro elements are they will be going to present. So here application of iron and magnesium to promote the synthesis of chlorophyll. If you are going to add it into the iron and magnesium into the plants, they can synthesize the chlorophyll pigments. Chlorophyll pigments will be doing synthesis. If the chlorophyll pigments are that in synthesis means automatically they can prepare the much amount of food metal by the process of photosynthesis. And option B 1C, frequent irrigation of the crop. Here we are going to supply the water into the plant frequently. So those are the plants will be going to get into the water normally and metabolical activity will be going to occur normally. But here in the hint they mention as a plant they will be going to premature yellowing of leaf. The premature yellowing of the leaf is a deficiency of some elements. Those some elements are macro and micro. So by the supplying of proper irrigation, that yield will not increase. And option C, chet it is treatment of plants with cytokinins along with the small dose of nitrogenous fertilizer. Here you will be get the confused in between the option A and option C. See, we have to increase the yield of plant and also we have to reduce the chlorosis disease. So chlorosis disease is occurred by the lack of some elements out of the some elements iron and magnesium also. So here when you are going to add it into the iron and magnesium elements, chlorophyll content will be going to increase. And here why we can compare the option A into the option C. If you are going to done the that is adding of cytokinins. Cytokinins are plant growth regulators. It can activate the or it can involve into the synthesis of chlorophyll pigments. Synthesis of chlorophyll pigments and also by adding the small dose of nitrogen in your plant body or all living organisms, one of the major biomolecule is nitrogen, and also nitrogen can act as a fertilizer. So if you are going to add it into the iron and magnesium, chlorophyll content only will be don't increase. If you are going to add it into the cytokinins, cytokinins are plant growth regulators. They can synthesize the chlorophyll pigments and also they are added into the small amount of nitrogenous as a fertilizer. So if you are going to add it into the cytokinins, chlorophyll pigments are synthesized and also they are added into the nitrogen molecules. Nitrogen molecule also one of the major component of chlorophyll. So you know that structure of chlorophyll pigments. In the center of the chlorophyll pigment, single magnesium element is surrounded by the 4 nitrogen atoms. So if you are added into the nitrogen, nitrogen also involved into the synthesis of chlorophyll pigments and also will be don't seen into the formation of nucleosides and the nucleotides. And this nitrogen is a major biomolecules out of the given different types of the elements. So here instead of the iron and manganese you are going to add it into the cytokinins and nitrogenous as a fertilizer. Chlorophyll content will be going to increase automatically. For the amount of the food material will be going to prepare much, and whenever the amount of food materials are they can prepare much, automatically yield will be going to increase. So that's the reason in between the A and the C. If the C option was not mentioned in the given option, you can choose as A. If they given as A and C, you have to give first preference for the C only because here they given as a cytokinins. Cytokinins they can involve into the synthesis of chlorophyll pigments and nitrogenous molecules are there used as a biomolecule and also they can act as a fertilizer. So and option D, removal of all yellow leaves and spraying the remaining green leaves with the 2,4,5-trichlorophenoxy acetic acid. So here, when you are going to remove the all yellow leaves, okay, you will be going to remove the all yellow leaves, and which of the leaves are they present? Those are all as a green leaves. To the green leaves, you are added into the 2,4,5-T, 2,4,5-trichlorophenoxy acetic acid. It is used as a weedicide. So if you are going to add it the 2,4,5-T, the green leaves also will be going to die. Automatically plants will be going to die, yield will not increase. So out of the given option, A and C are related. So out in these are the A and C, we have to give a first preference for the nitrogen molecules and also cytokinins are involved into the chlorophyll pigments. So answer is C. Instead of the C option, only A is there means you can choose as A also. And next question, which of the following statements is correct regarding to the absorption of minerals by plants? Here, plants are they can uptake the minerals from the soil. So at the time of the absorption of minerals from the soil, occur by two ways. That is passive absorption, active absorption. Passive absorption means without involvement of ATP energy. Active absorption means involvement or utilization of ATP energy. So here in the passive absorption, absorption of the elements from the soil into the plant body, that is root hair cells, occur by two ways. One, it is apoplast, and other one is symplast. So what happens in the apoplast? Elements will be don't enter from the soil into the intercellular space of root hair. This is the root hair. Here, these all of the elements will be going to come and store. So, movement of the elements are movement of these elements from the soil into the intercellular space of the root hair. That is called as apoplastic chain. Here, these are the apoplastic process is occur fast. And when the elements are there present here, intercellular space, these are the elements. These elements are they have to move from the intercellular spaces into the cytoplasm of a root cell. So here the plasma membrane it was present. So movement of the elements through the plasma membrane. Here a special type of the proteins are that involved. So this type of the movement of elements from the intercellular spaces into the cytoplasm of a cell it's called as a symplast. Apoplast and symplast. So apoplast process is fast than the symplast mechanism. So this is the passive absorption of elements, ATP energy will not utilize. And active absorption. So absorption of the elements takes place from the soil into the plant body, that is through the roots by the using of ATP energy. It is called as an active absorption. In these of the active absorption we are discussed as flux movement. So movement of the ions, that is into the root hair cells, it's called as a influx. Movement of the ions out into the root hair cells, that's called as a efflux. Influx and efflux are we are seen into the active absorption. See now, based upon this concept question, it is option A, ions move passive into outer surface. Yes, in the passive absorption, movement of the electrons are take place, movement of the elements are take place without using of ATP molecules from the soil into the root hair cells. And these are the passive absorption of elements. Apoplast and symplast, two steps are there. In that apoplast mechanism, elements are that move from the soil into the intercellular space. That's only we call as outer surface of the root hair cell. And option B, they given as outer space of metabolic process. Here the metabolic process means ATP energy is utilized. Outer space of metabolic phase has no free exchange of ions. Here metabolic energy is utilized means it is not belongs into the passive absorption, it belongs into the active absorption. And option C, outer space of metabolic phase is formed cell membranes, cytoplasm and other structures of living cells. Here also they can mention into the metabolic phase. Here metabolic phase it is represented as using of ATP energy. That ATP energy used means it is not considered as a passive absorption. And option D they given as outer space of metabolic phase. Outer space of metabolic phase is area of symplast. Here also in the D option, C option and the B option. These are the three options of B, C, D. They given as two types of the movements as that is passive absorption and active absorption. So outer space, the word it will be drawn to used for the passive absorption only. Outer space and the inner space. So and here active absorption and passive absorption both are they given into the option. So that's why we cannot consider into the right option for the absorption of elements. See option 1 or option A, ions move passive into the outer space. In these are the option A only they mention about the passive absorption, and these are the passive absorption that is mentioned as apoplastic way only. So, but in the B option, outer space, that outer space belongs in the passive absorption. And metabolic phase, metabolic phase means ATP energy is utilized, and in the same outer space of metabolic phase, here also passive absorption and using of ATP energy. So both are there. Here also outer phase and the metabolic phase they mentioned. So in these are the B, C, D options are they belongs into the both passive and active absorption. But in the A option only, they mention into the passive absorption, that to apoplast mechanism. By the elimination method, we can choose as a answer A. And next question, which of the following is a part of cytochromes? So cytochromes are the specialized structure of chlorophyll pigments. So at the time of photosynthesis in that cyclic and non-cyclic electron transport system. Intermediate components are their presence. Cytochromes. Cytochromes also light as a chlorophyll pigments only. These are the carrier molecules to transfer the electrons. So you know that in the photosynthesis and the respiration, that cytochromes are that can play a key role. These are the cytochromes are they made up of by. Options are option A, magnesium. Magnesiums are used for the synthesis of chlorophyll pigment because the chlorophyll pigments are made up of by the center single magnesium element. And zinc element, it is used into the activation of enzymes, alcohol dehydrogenase enzymes, like. And iron, here the iron molecules they are made up of by the cytochromes are made up of by the presence of iron molecules. And calcium elements are majorly used for the synthesis of cell wall as middle lamellum, and also calcium is used for the formation of spindle fiber. So cytochromes are then prepared, or cytochromes are made up of by the presence of iron and sulfur molecules. Answer C. Next question. The essential elements which are required for the plants in large amount are called dash, and those required in very small amount by the plants are called as. Here they are discussed as a elements available in a nature as a more than 105. So out of these 105 elements, some of the elements are they required for the plant growth and development. So which of the elements are they required for the plant growth and development? Instead of that elements, plant growth and development will not take place. Out of the 105 elements, 7 elements are we call as essential. So in these are the 17 essential elements are again grouped as a macro and micronutrients. So based upon the utilization of that quantity, which of the elements are they required more than 10 millimoles per kg? 10 millimoles kg inverse. Which of the plants are they required much elements more than 10 millimoles per kg inverse? Such type of elements all are we call as a macronutrients or macro elements. In a dry matter, in the same way, which are the elements are they required for the plant growth and development less than 10 millimoles per kg inverse? Such type of the plants, such type of the elements we call as a micro elements. Macro elements and micro elements, these are the macro more than 10 millimoles per kg inverse in dry matter elements are required for the plant growth and development, and micro elements are less than 10 millimoles per kg inverse in a dry matter elements are required for the plant growth and development. So these both are the macro and micro, we are combined together will be written as 17 elements. That 17 we call as a essential elements. So now the essential elements which are required by the plants in large amounts called as a macro elements. This here we can consider as a macro, and those are required small amount by the plants are called dash, here it called as a micro element. So first macronutrients, and next it is a micronutrients. So option A, micronutrients are they given here. Here it should be more than 10 millimoles per kg inverse elements are they required for the plant growth and development. So that's what we call as a macro. So out of the given 4 options, first should be present as a macro. They given as a macro here. And instead of this macro, in the B option they given as a bulk elements. Bulk elements means group of elements. Here macro elements and the bulk elements are also group of elements, but all of the bulk elements are we are not considering as a macro. So that's why some of the specific elements only they required for the plant growth and development more than 10 millimoles per kg inverse. So those type of elements are we call as a macro elements. And next is micronutrients or micro elements. And next question, identify the characteristic features of lead hemoglobin and select the correct option. Here lead hemoglobin pigments are the specialized pigments we are seen into the root nodules. So these are the root nodules of leguminous plants. So they can show the taproot system. So in these are the leguminous plants, root nodules are the presence in the root nodules by the entering of rhizobium bacteria. Here the rhizobium bacteria will be present. The rhizobium bacteria is covered or protected by the presence of a specialized structures. That specialized structures are present in pink in color. That pink colored pigments are we call as leghemoglobin pigments. So function of these leghemoglobin pigments and concept of these rhizobium bacteria in the plants of leguminous plants. That rhizobium bacteria was present in a soil can enter into the plant body, it can form into a root nodules. The root nodules contain of the leguminous rhizobium bacteria that can fit into the atmospheric nitrogen. So nitrogen fixation is occurred by the presence of rhizobium bacteria. These both are the bacteria and the plant body that can show the symbiotic association, symbiotic relationship. So based upon the leghemoglobin pigments concept, which is the correct option we have to identify. Option 1, it is a pinkish colored pigment present in a root nodule. Just these are the leghemoglobin pigments are the specialized pink color pigments are we are seen into the nodules of leguminous plants. And option 2, it helps in nodule formation. Here rhizobium bacteria is protected by the this leghemoglobin pigment. So here the leghemoglobin pigments are will not involve into the formation of root nodules. The root nodules are they are formed by the entry of rhizobium bacteria. Means option 2 it's a incorrect. And option 3, it protects the nitrogen fixing enzyme nitrogenase from the oxygen. You know that fixation of atmospheric nitrogen by the presence of specialized enzyme. That enzyme it is nitrogenase. That nitrogenase enzyme here we are seen in rhizobium bacteria. So these nitrogenase enzyme it can sensitive into the molecular oxygen. If the molecular oxygen is available, nitrogenase enzyme is inactive. If the molecular oxygen is unavailable, nitrogenase enzyme is active that fits the atmospheric nitrogen. So now you see this nitrogenase enzyme is activated in the absence of oxygen. So to prevent the availability of oxygen. So these rhizobium bacteria or nitrogenase enzyme is protected by the leghemoglobin pigments. That leghemoglobin pigments are provided into the anaerobic conditions into rhizobium bacteria or nitrogenase enzyme. At the time only that nitrogenase enzyme is active, atmospheric nitrogen is converted into the soil nitrogen. Option 3 is correct only. And option 4 they given as it is a source of energy. Yes, nitrogenase enzyme and the leghemoglobin pigments. Leghemoglobin pigment is not a source of energy. The source of energy it is ATP only. ATP synthesizing take place in a respiration, not by the presence of leghemoglobin pigment. So out of the given options, which are the options are they related or they are corrected into the leghemoglobin pigments? Option 1, it is a pink color pigment present in a root nodules. Option 1 it's correct. And also the option 3, it protects the nitrogen fixing enzyme nitrogenase from the oxygen. Oxygen, these leghemoglobin pigments are they giving as scavenger into the molecular oxygen. So option 1 and 3 are the correct statements. So answer B. And next question, zinc, molybdenum, iron, copper. So zinc, molybdenum, iron, copper are the option A trace elements, non-essential elements, macronutrients, and none of these. So non-essential elements and none of these eliminate and remain as a trace elements and macronutrients. Trace elements are we are also called as a micronutrients, and macronutrients are which are the elements are that can utilize the plant growth and development more than 10 millimoles per kg. So now here these are the zinc, molybdenum, iron, copper, nickel, boron, chlorine. Zinc, molybdenum, iron, copper, chlorine, boron. These are the elements we call as a trace elements are also called as a micronutrients. So answer it is a trace elements or micronutrients.